Oops, that helps. Okay, so cholesterol is going to be a component that you use for certain types of hormones. All right, and there's other functions for cholesterol also. Helps in making bile salts, and we know that bile salts, all right, are key to fat metabolism, all right, in our bodies because it helps with the process of emulsification, all right, is when we're trying to break down those fats, all right, in the small intestine. Okay, so we need cholesterol, and that's why I tell people, and they look at me like I'm crazy, all right, but cholesterol is actually good, but obviously too much of anything is never really a good thing, okay? But cholesterol, you should take that into your diet, all right, in moderation, all right? So when we talk about, all right, making cholesterol, all right, the cells that are responsible, or the organ that's responsible, is the liver. The liver is going to help to make the cholesterol, okay, specifically the hepatocytes, okay? So we have to start off with the building block for cholesterol, and that building block is acetyl-CoA, okay? And it's a 2-carbon unit, and it's found, all right, in our triglycerides, our fatty acids, all right, when we were breaking them down, and that's essentially what we do with triglyceride. We snap off fatty acids off of the glycerol backbone there, and then we start to just chop off the carbons until we're left, all right, with the last 2 carbons, all right, that 2-carbon unit, which is acetyl-CoA. And how we do that is through that process of beta oxidation. I'll show you a picture here, all right, on how we do that. And we discussed that roughly, you know, I didn't put it in so many terms when we were talking about in chapter 26 in digestion, all right, on how this is occurring. We did see how you have that, all right, that lingual lipase, all right, that salivary lipase, which starts to break down the fatty acids, all right, or excuse me, the fats in your mouth, all right, how it starts to cleave off, all right, one fatty acid off of your triglyceride, leaving you with a diglyceride, okay? And then how we get to the small intestine, all right, then we start to cleave off another fatty acid, okay? So we're going to start off with that acetyl-CoA, and then we need this very important enzyme, HMG-CoA reductase. This should actually be a capital A here, okay? Okay, so HMG-CoA reductase is that enzyme that's going to catalyze, and catalyze means it's going to kick it off. All right, it's like if you've ever done track and field, when they shoot the gun, that's the catalyst to the start of the race. All right, this enzyme is going to be the catalyst. All right, we're starting off with our acetyl-CoA. All right, that's our most basic structure. Then we're going to start to make cholesterol from that. But in order to do that, we need that HMG-CoA reductase, all right, to catalyze that chemical reaction, and this is what we're going to wind up with. All right, cholesterol. Okay, so everyone varies in how much cholesterol they can make. Some people have, you hear, oh, I've got good genes, you know, and some folks will have high levels of a certain type of cholesterol in their body, which they deem the good cholesterol, which we'll talk about here in a little bit. And then they have the other kind of cholesterol, which they call the bad cholesterol, which we'll talk about it, all right? But keep in mind, all right, when we're making cholesterol, it's going to vary from person to person here, all right? But if you think about it, and this makes sense, all right, when you take in a lot of cholesterol through your diet, your body doesn't have to make that much, okay? So a high cholesterol intake in your diet, all right, will mean a low amount of cholesterol being made by the liver, and vice versa. Okay, if you don't have a lot of cholesterol in your diet, all right, especially if you're eating no meats and you're mainly eating plant sources, but those plant sources happen to be poor in cholesterol, then your hepatocytes are going to increase the production of cholesterol depending on what your dietary intake is. Okay, so now we've made our cholesterol. All right, so let's talk about what happens now with that cholesterol after we've made it. I've got this awesome picture here, so we're going to talk about all the stuff that happens here in this picture. I'll zoom it in and everything. All right, so what we're going to see is once that cholesterol is made, we're going to release it into the blood. And we're going to release it into the blood as what we call the very low density lipoprotein, otherwise known as VLDLs. Okay, so once we make our cholesterol, if we want to send it out, all right, to other cells, all right, we have to package it up. We can't just send it out there in the harsh environment of your blood. Okay, so we're going to slap a bunch of proteins around it. All right, and we talked about this. All right, when we were discussing our fat, when we break down our fats in the digestion, how when we bring the triglycerides or the broken down components of triglycerides, when we bring in our fatty acids into the individual cells that line the digestive tract, we then make them and recreate them back into triglycerides. All right, and then we're going to send those triglycerides either into the lymphatic system, most likely, but some will go into the bloodstream. Well, we just can't send it out there, so we package those triglycerides up into what we call VLDLs. Okay, so those are our very low density lipoproteins. The key word here is proteins. We're just going to coat whatever we're making or whatever we're sending out of the cell with a bunch of proteins. Okay, so that's one way, or one option that happens to cholesterol after it's made. The other, and I've mentioned this before, is we're going to incorporate it into our bile salts. Okay, so we have these bile salts, and we know their purpose because they help to break down fats through the digestive process. But guess what? We need cholesterol to help to make these. Okay, so the liver will help to use the cholesterol that it's produced or whatever you've ingested through your diet, and it'll make the bile salts with them. Now, most of those bile salts get recycled, approximately 90%, but 10% gets lost in the feces. And this is where I mentioned last time, I think it was either in the video or in class, where I talked about the drugs that we have developed to help increase the excretion of bile salts with people that have high cholesterol. Okay, so you can increase the amount of bile salts from 10% in the feces to about 20, 30%. All right, but that forces the liver now to take cholesterol from the blood to make more bile salts. So it helps to lower your cholesterol levels. All right, some of those drugs don't have very nice side effects. You can get what we call fatty stools. Fatty stools are not the most pleasant to be around. All right, so that's one of the side effects. But we have better classifications of drugs now. We have the statin drugs and several other different types. Okay, but so these are our two choices. We'll either get rid of the cholesterol as VLDLs and send it out into the blood, or we will incorporate them into bile salts. So let's take this cholesterol that we made, and let's get rid of it, liver. Okay, so we're going to shoot it out into our blood. Okay, and here, whoops. All right, here's our hepatocyte. Okay, so in order to make cholesterol, we need acetyl-CoA. Where do we get it from? All right, our fatty acid through the process of beta oxidation. You remember what a fatty acid is? It's just a long chain of carbon and hydrogen molecules, all right, just strung together. And they vary in length, depending if it's a short chain, long chain, medium chain. All right, and then we just start to cleave off, all right, carbons. We pop them off, all right, through this process of beta oxidation till we get down to the final two carbons here. All right, that molecule is known as acetyl-CoA. Okay, and that's where we're going to start with the creation of our cholesterol molecule. All right, with the help of HMG-CoA reductase. All right, you need to know that. I can almost guarantee you'll be asked a question on that on the test in regards to HMG-CoA reductase. All right, in regards to the synthesis of cholesterol. Okay. So we have that enzyme that kicks off the party, okay? Now it varies, all right, depending on your dietary level of cholesterol. If it's low, then the synthesis of cholesterol in your liver will be high. It's that inverse relationship type of relationship that we got going on. I said relationship twice, all right? So then what we're going to see now, we have our cholesterol molecule, okay? So now we can have two options, okay? We can incorporate it into the bile salts. That's cool. All right, so as you know, the liver makes the bile, all right, it exports it to the gallbladder. The gallbladder will concentrate it and store it until you have a fatty-rich meal, and then the bile salts will be excreted in the bile or into the small intestine. Okay, so that's one route that it can go. The other route is to shoot it out into the blood. Okay, but we're going to do that, all right, if we're going to do that, we're going to package it up as our VLDS. Okay, our very low density lipids all right, we're going to use that to coat this puppy, all right, and we're going to send it out, and we're going to talk about this here in a bit, all right, what happens to those lipids, those fats, all right, as they're traveling through our bloodstream here, okay? All right, now this might interest some of you, because if you've ever looked at your blood panel, if you've ever gone to the doctors, and I know that most of you are very young, so you're not probably too concerned about your cholesterol levels and your good cholesterol versus bad cholesterol, but if they ever do a lipid panel on you, okay, if they're concerned about the functionality of your liver, all right, part of it is they're going to check your cholesterol, and they're going to check, all right, these specialized lipoproteins, all right, and these all have a role. And the three that we're going to talk about are the VLDLs, the LDLs, and the HDLs. I know, try it. It's not easy. Okay, so we're going to start off here by talking about the VLDLs. All right, and again, you look at the word lipofat, all right, protein. So this is going to be, all right, a specialized molecule that's going to be coated with proteins. All right, they're made in the liver and then sent out into the blood. Okay, and so this is a way for us to move around certain lipids. All right, for example, the triglycerides, we want to export those triglycerides because remember, the primary purpose for triglycerides is energy production. Triglyceride molecules are rich energy producers. Your body loves to use it. It would prefer to use it. It likes to use glucose, all right, but you just get a lot out of triglycerides. All right, that's why when you're in an energy-rich state, or what we call a fed state, okay, and all the energy demands for all this peripheral cells are needed, right, your body is going to then convert some of these macromolecules into triglycerides, which your body will store as fat. Okay, your body is always in that mode thinking, all right, you know what, I'm fed now, that's great, and I love it, but there could be a time later on when I might not have food. And I want to prepare myself. They're like, what do they call them? Preppers. Does anyone know the term prepper? All right. You see some people shaking their head, nodding their head up and down. Yeah, preppers are people that are preparing for some sort of event, whether it be a food shortage or whatever. So if you go into their basement, if they have a basement or wherever in their garage, they have all this food stocked away. All right, and they're prepping. They're getting ready. They have several years of food. All right, and so your body's like that. It's a prepper. All right, it's waiting for that opportunity, that time for when, all right, you know, energy sources are scarce, I need to prepare for it. And so that's what it's going to do, okay? So it's going to release, all right, these lipids as, all right, triglycerides packaged in these proteins, all right? So that's how we're going to, and we're going to walk you through this and kind of explain, all right? So the very low density lipoproteins are going to be the initial protein, all right, that gets kicked out of the liver. Okay, these triglycerides, all right, are all packaged up nicely, okay, with these very low density lipoproteins. Okay, now these VLDLs are going to head towards the peripheral tissues. Okay, and so when it arrives at these peripheral tissues here, all right, we are going to deliver the contents of our package. All right, those the triglycerides and our cholesterol here, we're going to deliver that, all right, our package to the peripheral tissues. Now, these tissues are wonderful because they put up these markers on the outside. And those are the LDL receptors. And they're these specialized receptors that are going to essentially catch these molecules as they're floating by and snag them. Okay, almost like a net. All right, and it'll snag them because they're attached to the plasma membrane here. And what will happen is the cell will then gobble it up through the process of endocytosis. And you remember the different parts of endocytosis is when you were going to take something that's on the external environment of the cell, and the cell is going to just kind of suck it up. All right, forms these pseudopods, they wrap around it, and they gobble it up. Phagocytosis is a form of endocytosis. All right, there's phagocytosis, cell eating, and there's penocytosis, cell drinking. All right, endocytosis is essentially taking something from the outside environment of the cell and bringing it to the internal environment. That's what happens here. Okay, so now we start to break down that molecule. Okay, if there's cholesterol present, awesome. Guess what? We're going to take that cholesterol and boop, we're going to put it into our cell membrane. cholesterol does what for the cell membrane? Do y'all remember? If you watched the video, I did mention it. It adds stability to the plasma membrane during temperature extremes. Okay, it makes the plasma membrane able to handle temperature extremes. All right, makes it more, I'm not going to say rigid, but more sturdy. Okay, if you're in a very hot or cold environment, it helps with that. Okay, also, all right, if this is a specialized cell that is of endocrine origin, all right, it helps to make cholesterol-based, which are mainly steroids, all right, steroid hormones. Okay, so we'll see that that cholesterol can be used here, all right, for the production of steroids. Okay, so cholesterol goes a long way with a lot of different things. All right, so if you go back here, you can see that cholesterol molecule here. All right, I know it doesn't really seem like much. All right, but when you break it down, and I talked about this in the first video here. All right, these are all carbon rings, so there's tons of carbon in here. All right, and again, that's energy to be used. All right, and it's also, all right, a precursor when we're starting to make, all right, that's not the one I wanted, our hormones there. All right, when we're dealing with steroid hormones, and also if we're dealing with bile salts. Okay, so this is what, if you get a lipid panel done on your blood work, okay, this is the cholesterol that we refer to as bad cholesterol, okay? LDLs are considered to be the bad cholesterol. So if you have a high level of LDLs, that's not good, all right? Because it's assumed there's several different reasons for that, all right, as to the levels of the cholesterol floating in your bloodstream, all right? But if you have high levels of LDLs, then it tells us several things, but one of which is either your liver's making a lot of cholesterol for whatever reason, could be a genetic thing, okay, or you're taking a lot of cholesterol into your diet. So we try to manage that. All right, don't eat garbage. All right, don't eat, high fatty foods. All right, be more mindful. All right, and we'll see if that makes a difference. Here's the one that we want to have. We want to have this. The HDLs are what we consider to be the good cholesterol. Okay, and so these molecules, all right, are going to transport, all right, your cholesterol, your fats from the peripheral tissues back to the liver. Okay, so the LDLs are going to go to the peripheral tissues. All right, the HDLs are leaving the peripheral tissues and heading towards the liver. Okay, so and I'll walk you through this here. Okay, but these guys are the ones that you want to have in high numbers here. Okay, because basically think of them as a sponge. They're going to go around and just start sucking up, all right, the cholesterol, the fats, all right. They're going to go around and pick up triglycerides and fill up, okay, on their way from the peripheral tissue back to the liver here, okay. All right, let me show you a good picture. I like this. This is the story here of lipid transport in our body. This will help you a little bit. All right, so I'm not going to start with the HDL. I'm going to start here, all right, with the VLDLs. Zoom out a little bit. Okay, so here's our hepatocyte here, okay? And so what your hepatocyte has done, okay, it has incorporated the cholesterol and any triglycerides, and it starts to package it up into these molecules, into the VLDL, the very low density lipoproteins, okay? And it exports them out into your bloodstream, okay? So they're going to float throughout the bloodstream, and they're going to travel to different types of peripheral tissue, okay? In some cases here, you're going to see when we're dealing with the VLDLs, they're going to release triglycerides into adipose tissue. Well, that's good, right? That's what I was telling you about. Here's your body going through that prepper motion, okay? It is actually going to take those triglycerides and store them, all right, in the adipose tissue for a rainy day, for when you need it, okay? When it does that, okay, now it's gone from being a VLDL to an LDL. Okay, so you can see here it looks like it's full. Now it's starting to empty out a little bit. Okay, then it's going to circulate throughout the bloodstream looking for cells, all right, that are going to have, all right, the LDL receptors on them. Okay, and those cells will vary, all right, from hormone-producing cells to some cells that just need, all right, needs cholesterol to help improve its plasma membrane. Okay, so it's going to circulate around. It's going to find a cell, all right. It's going to connect to the LDL receptor, all right. Then the cell is going to undergo endocytosis. It's going to pull that LDL into the internal environment of the cell, and then it's going to incorporate that cholesterol into the plasma membrane or use it for steroid hormone synthesis. Okay. So meanwhile, okay, now we have these guys here are HDLs. All right. So they go from the peripheral tissue back to the liver. So you can see here, here's an empty HDL, and it's just going to circulate through your peripheral circulatory system, looking for, all right, cholesterol to soak up as it's circulating through your bloodstream. That's what it does. That's why you want to have a high number of HDLs, because they're going to go around and start absorbing that cholesterol. Once it gets full here, okay, it's got its full fill, it's got its fill of cholesterol and triglycerides. Then it heads back to the liver, all right, where it empties out its cholesterol and triglycerides. All right, so your hepatocytes can now package those up if they need be, all right, into those VLDLs, or maybe it needs to make some bile salts. So it'll take some of that cholesterol that it got from the HDL and use some of that cholesterol to make bile salts. So just think of the HDLs as just circulating through your bloodstream and just looking for triglycerides and cholesterol to soak up, all right, to bring back to the liver for whatever. So you can see why you want to have high numbers of HDLs. That's why some doctors aren't concerned, all right, if your cholesterol is like usually anything above 200, all right, is considered to be bad, right? So about 210, 220. But some docs won't really be as concerned if you have cholesterol that's around 205 or 210 if your HDLs are pretty high, because the HDLs, all right, are going to help to keep, all right, that cholesterol that is floating around at a manageable level. Okay, so we'll see that from time to time. All right, questions about any of that? Good so far? I forgot to fill up my water, so I have to sit here with dry mouth. That's just how it is. All right. Okay. So this is kind of like what I just mentioned. All right, I jumped ahead of myself. Okay. So if we see high levels of LDLs or high levels of total cholesterol, we're thinking cardiovascular disease. That's a problem. You guys have heard of, you folks have heard of atherosclerosis, blockages of arteries, all right? That's what folks get concerned about because, all right, a lot of the times when we're dealing with atherosclerosis, all right, there is some sort of physical injury, all right, to the blood vessel itself. And when you get that physical injury to the blood vessel, okay, usually propagated by some sort of inflammation or some sort of trauma, now you have exposed endothelium, because you know the lining of the blood vessels is lined by endothelium. And then the layer underneath there, okay, when we get to the tunica media, all right, depending if we're dealing with an artery, okay, once that area gets exposed, you have those chemical markers, and I talked about it before, the von Willebrand factors there, all right? But you start to get almost the clotting cascade start to occur, which is normal. But the problem is, if you have high levels of fat and cholesterol floating through your blood, it will get lodged into that damaged area there. Okay, there's a big process to it, but essentially that's where you get what we call the plaque. All right, that where it starts to kind of, normally the lumen of your blood vessel is like that. Okay, but when you get a little damage here, all right, things start to fill up in that damaged area and you start to get this. All right, you get like this fatty plaque. It's just filled with fat and cholesterol and red blood cells, and it's like a bloody scab, all right, loaded with fat. So now, remember what we said about our blood flow. We want the blood flow to be nice and laminar. Just wants to kind of go down nicely. But now look what we've done. We put a bump in the road, and that causes turbulent blood flow, which can then cause more damage to the blood vessel, which can now make the situation worse. And over time, more stuff will accumulate here, and we start to get a blockage. Well, guess where we see that blockage? Coronary blood vessels of the heart. That ain't cool. That's a problem. Okay? So that's what we start to see here. I know I went off on a tangent. I apologize. Not really. Okay, so values that are acceptable, anything below 200, obviously, we'd like to keep you way below that, 180, 170, okay? But as we start to approach 190, then we start to get concerned. All right, anything over 200, that means high cholesterol. Keep that in mind. All right, milligrams to deciliters. All right, LDLs, those are the bad cholesterol. All right, that is what we see in that fatty plaque that I was just drawing in that picture there. All right, those LDLs. The HDLs, remember, their job is to go around the bloodstream and soak up, all right, the triglycerides and the cholesterol. We love this stuff. It's the good cholesterol. So it's going to clean off, all right, the arterial wall, okay? So you want to see high levels of HDLs, low levels of LDLs, and obviously low levels of total cholesterol, all right? That's ideal. All right, has anyone here ever gotten their lipid panel done? No? I wouldn't expect because you're all pretty young. But if you have family, my cholesterol is 290. Really? Do you know what your HDLs are, Casey? Triglycerides are 202. All right. This is where I would ask you questions, but I can't because of personal medical information and whatnot. I can't remember what. Okay, yeah, that's usually the one that I go to. And my cholesterol has not been over 200, but I was told that my HDLs were pretty good and then my LDLs were normal. And I've had it done. I've had my blood lipid panels done, I don't know, lots of times. I used to do, oh, really? All right. Well, when I see you on Wednesday, we'll talk. We'll talk. I've got questions. Just remind me. All right. Let's jump into, you're funny. All right. What we're looking at here, because now we're going to move on to another part of this topic of metabolism. And I really want to talk to you about the importance of the liver. Now, we know the importance of the liver when we're dealing with it as an accessory organ of the digestive system. It makes bile. All right, that's cool. But the liver does so many other things. It makes tons of proteins for whatever different systems in your body. But you can see here, all right, look at all the different roles that it plays. Carbohydrate metabolism. Well, we know that's where gluconeogenesis occurs. So we have a non-carbohydrate source to make glucose. That's cool. The liver takes care of that. And actually, we talk about it mainly with the liver, but the kidney can actually get involved in that. All right. But for the purpose of this class, all right, the liver, okay, for gluconeogenesis. The kidney plays a very mild role if it has to. But usually the liver can handle that. All right. Protein metabolism. Remember albumin? Remember that one protein I was talking about when we were talking about all the different types of ingredients in our blood plasma and our blood? All right, well, albumin is the most prominent protein. And again, its job, all right, well, not its job, but one of the characteristics of albumin is that it increases the oncotic pressure. Remember that oncotic pressure is the pull, all right, the pull of things into the bloodstream, all right? And proteins exert an oncotic pressure, all right? So we see that, and plus all the different proteins that are also made, all right, for clotting, all right? Those proteins are also made in, all right, the liver, okay? Lipid metabolism, we just talked about it. We talked about its role in lipid metabolism here, okay? And how it offers to transport those lipids through the process of exporting VLDLs, excuse me, and then with the role of HDLs with the liver, okay? And then you probably don't remember, all right, but back in chapter 4, we talked about the different structures in cells, and we talked about the endoplasmic reticulum, and there was a difference. There's a rough endoplasmic reticulum and a smooth endoplasmic reticulum. The rough endoplasmic reticulum primarily is involved in protein synthesis. Smooth endoplasmic reticulum is this: drug detoxification and alcohol detoxification. So the hepatocytes in your liver are loaded with smooth endoplasmic reticulum, and that's the job. Detoxify drugs. What does that mean? Well, you have certain enzymes that break down drugs. And in fact, a lot of the drugs that, if you're on any prescription drugs, I can almost guarantee you that most of those prescription drugs that you take, if you take any, rely on certain enzymes in your body to be present to break that drug down. Because a lot of the times when you take a drug, it's in the inactive form, and your body needs to activate it. We call that first pass, second pass metabolism. Okay? So we'll see that, all right, with the liver. Some of those enzymes are in the liver. And if they're not in the liver, we're looking to the kidney. Okay? You get to learn about all that stuff if you're going to take pharmacology. Fun stuff. All right? Okay, so proteins. Our proteins, this is the, this is the last of the macromolecules for this chapter. All right? So I can't say enough about proteins. Proteins, and there's, on the next slide here, we're going to see all the functions, and this is, I use the same exact slide, this next slide here, in Bio 210. So some of it might look familiar to you, all right? But you have to understand, these molecules are the most functionally diverse out of all the molecules, all right, when we talk about the macromolecules, all right? Carbohydrates, yeah, sure. Lipids, a little bit more, but proteins, and when you take a look, and I'll just quickly flash it at you, look at all these functions that proteins are utilized. Obviously, catalysts, for, they're enzymes, all right? Defense, antibodies. Anybody say antibodies? All right, we're involved with that. Transport, hello. When we're talking about any channels, all right, or pumps in the plasma membrane of your cells, those are proteins. Those are proteins. All right, for structural support, all right, when we're talking about the cell cytoskeleton, we definitely get involved in that. All right, proteins. Movement, okay, well, think about it. Actin myosin, all right, the contractile filaments of the sarcomeres, all right, are going to be those protein molecules there. All right, so you can see, look, these are just tons of functions for proteins. So it only makes sense, all right, that are the most functionally diverse here. Okay, well, with anything, all right, over time, all right, wear and tear, these things will break down. So we will have to constantly replace them as they break down. A lot of that has to do with maybe the type of protein and its function. Pumps, all right, channels all the time, all right, things break down, we got to repair them. But as we get older, all right, as we age, all right, we'll see that occur, all right? So you can see how when we're discussing proteins, how they play a role in almost everything that we do. So what's the building block of proteins? Do y'all remember?
Amino acids.
Yay, AAs, all right, amino acids, okay? And so we have 20 amino acids in the body, okay, that we're going to discuss. 8 are what we call essential, all right, which means you have to take those in through your diet. You can't get those, the body can't make that, all right? And then you have 12, all right, that can be made in the body, and those are called non-essential, all right? So not like essential workers and non-essential workers, it's not like that, all right? Basically, when we break down amino acids, all right, there's the essential, which is you have to take those in through your diet, and then you have the non-essential, all right, which are brought in, or that your body can make, okay? So I'll just throw this slide back up there real quick here, just so you can kind of see all the different functions for our proteins, all right? Regulation all the time. Chemical reactions in your body are going to be governed, all right, by the proteins there, all right? And then storage, all right, when we're discussing how things are going to be stored, and especially when we're dealing with, all right, the metabolism of glycogen production and glucose metabolism, all right, plays a role. Even when we're discussing how triglycerides and cholesterol get stored in your adipocytes, proteins play a huge role in all that. All right, so like I said, building block for proteins are amino acids, all right? And basically what we're doing is we're just going to start sticking amino acids, all right, onto one another, all right? Those are our monomers, all right? And if we get 200 or more amino acids, that's a protein, okay? And I'll show you here. I know I'm jumping ahead here. I'll come right back to this slide, I promise you, all right? So a protein with 200 or more amino acids, or a structure with 200 or more amino acids, that's a protein, okay? And then our polypeptides are 21 to 199 amino acids. These are very specific numbers, by the way. We learned that back in chapter 2. Yep. And then from 3 to 20. So when we're doing digestion, we're starting with this molecule here. All right. So mechanical digestion starts to break proteins down into polypeptides. Okay. We start to break that down, chew them up or whatever, and some chemical digestion will help with that. Ideally, by the time we get to the small intestine, we want to be after the stomach because of pepsinogen, all right, and hydrochloric acid will definitely break these polypeptides up into oligopeptides. All right, so at that point, we're dealing with 3 to 20 amino acid chains. All right, and then when we get into the small intestine, that's where our brush border enzymes are going to start to really chop those oligopeptides up into hopefully amino acids or dipeptides, and then eventually we can chop those dipeptides up into amino acids so we can absorb them. But we already learned about all that stuff. Let me jump back here. Okay. So one of the things that you need to know on our proteins, okay? On one end of the protein, all right, you're going to have an amine group. An amine group includes a nitrogen, all right, and it's got hydrogens bonded to it. That's the amine group, okay? Think of ammonia, all right? Then on the far end, we have the carboxyl group, all right? These are both functional groups, all right, and they're also polar, all right? Which means when we say polar, that means one end of your protein molecule will have an amine group, all right? The N terminal, they call it, and then at the other end, you've got the carboxyl group, that's the C terminal. All right, that's how we defer polarity, all right, to our molecule, okay? So this is what we're going to see. Now, when we have an amino acid, we're going to have a carbon unit in the center, and then that carbon unit will be, all right, bonded to an amine group on one side and the carboxyl group on the other. Okay, that's all we're saying. And then that carbon will also have a hydrogen element attached to it. And then it'll have what we call the R group, which is a specialized group, and that's how we're able to determine amino acids from one another. All right, that side chain, that R group, will be different depending on what type of amino acid we're dealing with. All right, so we'll have our amine group at one side, our carboxyl group at the other. All right, then in the center we'll have our carbon with a hydrogen attached to it, and then our R group, which is just a specialized side chain. All right, that distinguishes one amino acid from another. All right, so if you look here, that's what we're looking at. Here's our amino acid. Okay, so here's our centralized carbon with the hydrogen attached to it. Here's our R group. And it varies depending on what amino acids we have. Then we have our amine group. Here's our nitrogen with the hydrogen attached to it. And then our carboxylic group here, all right, which is going to have, all right, our carbon group with a double bond to the oxygen and our hydroxide group. Okay, pay attention to that because, all right, when we make a peptide, all right, that's 2 amino acids or 2 or more amino acids that are going to be attached to one another. All right, this is going to come off and this is going to come off. Okay, H and then OH to make water. I'll describe that in a second. All right, because that's what we're going to see here. When we take 2 amino acids and we stick them together and we get a dipeptide. Okay. And so as a result of that dipeptide, all right, we get water. It's called dehydration synthesis. Remember that from the video? Okay, dehydration synthesis, all right, is a catabolic reaction in which the byproduct of that reaction, all right, you're going to start with 2 smaller units that make a bigger molecule, and as a result of that, we get water. Dehydration, okay, water comes out of that. Okay, I'm jumping ahead. I'm getting ahead of myself. All right, whoops. There we go. Okay, so if you didn't already know this, you should know this. The name of the bond between the amino acids is called a peptide bond. All right, this is the bond that's formed during dehydration synthesis. All right, this is also the bond that is broken during hydrolysis when we start to break. That's what we're going to see, all right, during digestion. Okay, chemical digestion in the stomach, in the small intestine. All right, that hydrolysis is when we start to snap these peptide bonds down, all right, in between the amino acids. All right, and our goal is to get individual amino acids once again. Okay, but as a result of the dehydration synthesis reaction, when we're taking two individual amino acids and combining them to one another, all right, one amino acid is going to lose the hydrogen off the amine group, and then the other amino acid is going to lose the hydroxyl group off the carboxylic end. Okay. All right. So here you can see. All right. Here is our amino acid chain, just strung along. Now again, at one end is our amine group. All right. That's the N terminal. This is going to defer polarity to this structure. Okay. So you can't say that proteins are not polar. They are polar. Okay, and then at the far end here, the C terminal, that's the carboxylic acid. Now, if you remember back in chapter 12, I love to pull stuff back from stuff that you've learned before because it helps with the whole learning process. That's what I'm here to do, is to teach you, and you're here to learn. All right, so when you learn about the banana floating in the ocean, all right, how the neuron has a lot of potassium inside the cell and a lot of sodium and chloride outside the cell. And the inside of a resting neuron is negative. And the main reason, does anyone remember what the main reason why the inside of the cell is negative? What defers the inside its negativity? Two things. Inorganic phosphate, which is negatively charged, and proteins, negatively charged proteins. And the reason why is this here, the C terminal. That defers that negativity to that protein molecule. Okay, just in case you were wondering, I thought I'd tell you. Okay. All right, so here we are talking about our proteins. Keep in mind the numbers. An oligopeptide, 3 to 20 amino acids. Polypeptide, 21 to 199 amino acids. 200 or more, now we have a protein. Okay, so it's got to be big. All right, and then of course glycoproteins. Again, that's a protein with a carbohydrate group attached to it. We saw that before. We're going to talk about some glycoproteins here shortly when we get into the lab portion. All right, because we're going to talk about oocytes, which are the female eggs and the follicles. All right, we're going to talk about some more glycoproteins. All right. But these are going to, the example we saw when we were doing blood typing, all right, ABO. Okay, that's a glycoprotein. All right, so here you can see all the different types of amino acids, organic amino acids that we're going to see in our body here. Okay, so they have, and this is basically, I'll zoom in a little bit. Again, you don't have to memorize all these. Okay, here you can see, check it out. These are the side chains here. They're just different. And these side chains defer, all right, certain functionality, all right, to these amino acid groups here. All right, but they all have the same. Here's your amine terminal, your N terminal. Here's your C terminal, or the carboxylic end here. Okay. All right. Last topic of this chapter, or chapters, I should say. And that is going to be how we make energy. Okay, so there's a reason why you're eating, all right? You're eating to help to restore, all right, nutrients that may have been lost, all right, through certain processes, all right, of metabolism, or you may have damaged certain structures, okay? So now we're going to discuss how we make ATP. And there's going to be 4, all right, parts to this ATP production. Okay, or stages here. All right, the first one we've talked about is glycolysis. So we're going to take one molecule of glucose and we're going to break it down and try to get some ATP out of that. This occurs, all right, in the cytoplasm. You need to know this. Not outside the cell, all right, but inside the cell. Okay, but outside the mitochondria. Important that you know that. So one molecule of ATP is going to yield 2 pyruvate molecules. Okay? That process is called oxidation. Okay, so when you're undergoing glycolysis, you're breaking down one molecule of glucose via oxidation into 2 pyruvate molecules. And as a result of that, we get 2 ATP. All right, not bad, not bad, okay, but not good. Okay, as a result, also, we're going to get 2 NADH. We'll review that when we get to the electron transport chain. That H becomes important because that's going to help to give us some energy later on. Okay, so if we have oxygen, cool. All right, now we can now take those pyruvate molecules and bring them inside the mitochondria. But if we don't have, all right, oxygen, then we're going to wind up with lactate. Okay, so here's your glucose. All right, it's undergoing, all right, glycolysis here. All right, and it will yield us 2 pyruvate. All right, but if oxygen is not present, then we take a sharp turn, and instead of getting pyruvate, we go over here and we get lactate. That's not a problem though, guys. You know why? Because we can take that lactate and we can take it to the liver. And guess what the liver will do? It'll fall asleep. No, it's going to make glucose with it. All right, it'll convert that lactate into glucose through the process of gluconeogenesis. That's cool. All right, that's cool. All right, so now get rid of all that because we're not going to deal with that. We're going to deal with the fact that we do have oxygen. So we're going to move to the next step. All right, we're going to go to the intermediate stage. Okay, so now we've got the pyruvate. All right, now we're going to convert that pyruvate into, oh, wait a minute, I've seen this before, acetyl-CoA. Okay, all right, that was the beginning of cholesterol synthesis. All right, so what is acetyl-CoA? Well, that's what we're left with when we start to break down our fatty acids and we get down to the last 2 carbon group. All right, and so we call that acetyl-CoA. All right, so now we can undergo the process of what we call beta oxidation. That's the metabolism of fatty acids for energy. Okay, and we can whittle those fatty acids down all the way to this molecule of acetyl-CoA. Okay, and then once we have acetyl-CoA, we can get that into the mitochondria. All right. And we're going to start the 3rd step here. But the intermediate stage is going to give us some carbon dioxide. All right. We've seen that before. That's one of the waste respiratory gases, all right, that our red blood cells will pick up and take out of there. But also we got this guy again, NADH. It's another coenzyme. All right. And that's used for, we're going to talk about that when we get into the energy here on the next page. All right. So the intermediate stage takes us now to the citric acid cycles. Anyone ever heard of that? The Krebs cycle? It's not really referred to that as much anymore. It's more the citric acid cycle or the TCA. Did you go over that too? Briefly. I didn't get into the specifics of it in 210. I remember learning about it, but I might have been like, well, I could get into it a lot, but I don't. Here, this is because I talk about it here. Okay? So now our acetyl-CoA, all right, is going to be converted into many different what we call intermediates. All right, but citric acid is one of them. And it's going to go through what we call the citric acid cycle. It's this like wheel-like structure. All right, and we'll see, all right, acetyl-CoA get converted into several different compounds. And as a result of that, just one turn through, all right, that one turn through is going to yield more carbon dioxide. We're going to get some ATP from that. Cool, we'll get some energy. I like that. And then we get these 2 guys, FADH and NADH. Again, those are coenzymes. The important thing from both of these molecules are what are at the end here. These guys. Hydrogen. All right? Because we're going to take those hydrogens, and at the very end, it's like when you have a big night at the casino. Yeah, one big, I'm going to take my chips, and what do I do with my chips? I'm going to go cash them in. So guess what? These molecules are going to go cash in those hydrogens here at the ETC, or the electron transport chain, or electron transport system here. And this whole process, we're going to transfer hydrogens and electrons from these guys here, from these coenzymes here, and that process is going to create ATP. And so the name of that process, the whole process is called the electron transport system, but the chemical process that it undergoes is oxidative phosphorylation. Now don't worry, I got a cool picture to show you this. Okay? So generally, you're going to yield, all right, about, and depending on if all the ingredients are right and everything's correct, all right, your book says one glucose molecule will yield you 32 ATP, all right? But I've also seen it where it says that it'll also yield 36 ATP. Now, there's reasons for that, but we're going to go with the literature in the book as 32 ATP, all right? All right, so let's look at a picture here. I'm going to come back to this stuff real quick. All right, I just want to break down the general components of this and then we'll go through it step by step. All right, so here at the very top, glycolysis, step 1. All right, here's our glucose molecule. This happens in the cytoplasm, okay? The glucose molecule gets broken down into 2 molecules of pyruvate, okay? If there's no oxygen available, that pyruvate turns into lactate, okay? If oxygen is available, then we're going to move into our aerobic cellular respiration. We're going to get into the mitochondria here. As a result, all right, we will generate 2 ATP and an NADH coenzyme molecule. Remember that H, we need that H for later. That's going to give us, all right, some ATP in the electron transport chain. All right, come down here to the intermediate stage. All right, the pyruvates get converted into acetyl-CoA. Okay, so each pyruvate gets converted into 1 acetyl-CoA. As a result of that, we get carbon dioxide, no problem, and then another coenzyme, no problem. Okay, so acetyl-CoA will enter into the citric acid cycle. It'll take 1 turn, and as a result of that 1 turn, we'll get ATP, and we'll also get carbon dioxide byproduct, and then we get more, all right, coenzymes, NADH, FADH2. All of these coenzymes will come all the way down here to the electron transport system, okay? And you can see here, all right, in case I didn't mention this, we are now on the inside of the mitochondria, okay? The mitochondria have these double-walled structures. They're called cisternae, all right? And within these walls, you have these hydrogen pumps here, okay? And so, as a result, all right, when the hydrogens move off through these pumps, all right, and they cross over, it is going to undergo that oxidative phosphorylation. I won't get into the biochemistry of this because you don't need to know that for the test. But the point of it being, all right, when those hydrogens come off, it moves electrons over, and this is basically what we're going to see. We are going to see, all right, basically phosphate ions getting placed onto ADP, creating ATP. And as a result of that oxidative phosphorylation, hence the term phosphorylation, we're taking phosphate and we're slapping it on to a molecule, ADP, and creating ATP. And that's how we're getting our energy molecules. So 1 glucose molecule, all right, as long as oxygen is present, is going to yield us 32 ATP. Okay? All right, I'm going to come back to the rest of the stuff here on this slide. I just wanted to basically break down the 4 stages here. I want to just wrap this up because there's some stuff I want to talk to you about on the slide. All right, so hence, fats, okay, lipids. The most common or energy-rich lipid is going to be the triglycerides. Triglycerides are made up of that glycerol backbone and the fatty acids. Okay? So we can get, all right, these molecules to enter into that metabolism, all right? First of all, for glycerol, all right, we can get it into the glycolysis pathway. How do we do that? We ship it off to the liver. The liver drops it into gluconeogenesis, and it converts glycerol into glucose. Bam. Okay? Fatty acids, like I said with fatty acids, they'll undergo beta oxidation, all right? And we'll start chopping those carbons off. Do I have a picture? I probably don't have a picture of fatty acids. Anyways, they'll start chopping those carbons off until we're left with the acetyl-CoA, the last two carbons on our fatty acid molecule. And then we can drop that acetyl-CoA right there in the intermediate step, right into the citric acid cycle. Show you. Okay? So here you can see, here are triglycerides. So we can cleave off the glycerol backbone, and the glycerol backbone goes all the way to the liver. And the liver will convert that glycerol into glucose through gluconeogenesis and drop it into the glycolytic pathway here. And then, all right, we have our fatty acid chains, which we just start chopping off those carbons until we're left with the last two carbon molecules, which is acetyl-CoA. As long as oxygen is present, bam, we get right into the mitochondria, we go through the citric acid cycle. So there's a lot of different options here that your organs have to undergo, all right, depending on what you're dealing with. All right, we talked about carbohydrates, all right, and the importance of glucose. Now, we just talked about fats. Let's not forget about proteins, okay, proteins. So again, our proteins, we can still utilize, all right, to make ATP. But now what we're going to have to do here is, all right, we have to break those proteins down. Same thing, all right, we have to undergo some catabolism, all right, but we have to first remove the amine group. What is that? That's the NH2, right? It's a nitrogen with two hydrogens attached to it. All right, the process of that is called deamination. All right, starts in the liver, then that amine group gets sent off to the kidneys. No problem. Guess what? It's going to get dumped into your urine. All right, and that's what gives your urine that ammonia-like smell, because the amine group helps to contribute to ammonia. All right, so we're going to send the amine group here into the kidneys, which will be converted into urea, all right, which is one of the components of urine. So that will occur in the kidney. Okay, so once we've split off the amine group, perfect. What are we dealing with? We're dealing with, depending on what amino acids are present, all right, now we're going to deal with, all right, our carboxylic acid on there, but also what do we want? We want amino acids. We want amino acids. And it depends on which type of amino acid that we're dealing with, all right, which of the essential and non-essential amino acids. Point being is, all right, we can enter into this metabolic pathway either through glycolysis, all right, start at the beginning. Well, how does that happen? How do you think the amino acid can enter into glycolysis? What's the only thing that can enter into glycolysis? Glucose. Glucose. Glycolysis is the breaking down of glucose. So that means we have to convert the amino acid into glucose. Well, I know somebody that can do that. All right, the liver. The liver, through gluconeogenesis, will convert. All right, so we can go in through glycolysis. We can go through the intermediate stage or the citric acid cycle. Let's take a look on our diagram here. All right, so here's what happens up here in the liver. We have our proteins. They get broken down, all right, through the process of deamination, where we split off and get rid of the amine group, the NH2, which gets sent to the kidneys, all right, which you'll excrete in your urine, all right, it'll get converted into urea. All right, now our amino acids, all right, can be converted into keto acids, which can be converted into all right, through gluconeogenesis into glucose, all right, but we could also, all right, send in these keto acids through acetyl-CoA in the intermediate step, or we can drop them here right away into the citric acid cycle here. So you can see there's quite a few ways, all right, to get these different macromolecules, all right, introduced into metabolism here. Okay? And if you really like this stuff, take a nutrient, well, I'd say biochemistry class. Who here loves this stuff? No? I know. It can be boring to learn. No, and I understand that. And trust me, it can be somewhat overwhelming. Pardon? You do. You do. There's a lot of straight up memorization. And so once you lay the foundation, no, it's true. But when I first went to med school, I had to learn this. I had to learn all the 12 steps of the citric acid cycle. And so I had to be able to tell you the intermediate name. I forgot a lot of it. And then tell you exactly what enzymes were used to catalyze that. So that was quite a pain in the butt. So you guys only have to know the name of the cycle, all right, and what comes out of it, all right. All right, any questions about any of that? Anything jump to mind? It's a lot, I know. All right, let's do this. Let's take about a 15-minute break here. For you folks at home, all right, I will start lab early since we got done with lecture early, all right. This is on the test, this material here. This ends all the material for lecture test 4, okay? So chapter 26 and the metabolism information. All right, so let's take about a 15-minute break. We'll meet back here. I'll get, and I might even, oops,